Welcome to Let's Learn Plants. Today, we are diving into an explainer on proteins, the absolutely magnificent biological machines that are central to nearly all life processes. I'm so thrilled to guide you through this tutorial. We're going to break down the incredible architecture that allows these molecules to do virtually everything that keeps living systems functioning. Here is our roadmap for today's explainer. 1. The power of proteins. 2. Amino acids, building blocks. 3. Primary structure and backbone. 4. Secondary structure, folds. And finally, 5. Tertiary and quaternary assemblies. We'll be moving from the simplest building blocks all the way up to complex 3D structures. Section 1. The power of proteins. Let's kick things off by looking at their incredible versatility. Proteins perform an astonishing array of crucial functions. Think about it. From acting as enzymes that speed up essential chemical reactions to transporting and storing molecules like oxygen, they literally do it all. They provide structural support in our tissues, give us immune protection, generate movement in muscle cells, and even transmit nerve impulses. The wild part? All of these widely different functions are performed by linear polymers with highly specific defined structures. Section 2. Amino acids, building blocks. To understand how proteins can juggle all these different jobs, we really need to zoom in on their fundamental monomer units. Every single amino acid has the exact same basic chassis. You have a central alpha carbon bonded to 4 specific groups: an amino group, a carboxylic acid group, a simple hydrogen atom, and then the really important one, a distinctive R group, or side chain. It's this R group that gives each and every amino acid its unique identity and chemical personality. Now, because that central carbon is attached to 4 different groups, amino acids are chiral. Basically, that means they have left and right-handed versions known as L and D isomers. They're mirror images. But here is a fascinating quirk of biology. Biological proteins strictly utilize the L isomer. So if you ever find a D isomer out there, it for sure wasn't put there by standard cellular protein synthesis. Another really cool physical property is how they behave in water. At a neutral pH, these building blocks exist predominantly as Zwitter ions. What that means is they carry a positive charge on the amino group, which has gained a proton, and a negative charge on the carboxyl group, which has lost one, all at the exact same time. They're essentially tiny little dipolar magnets. So, nature builds every protein out of a standard set of 20 common amino acids, and we categorize them into 4 distinct groups based entirely on that unique R group we talked about earlier. We have the hydrophobic nonpolar amino acids. These guys hate water. We have polar amino acids that are neutral but have uneven charge distributions, making them water-loving. And then we have the highly hydrophilic, positively charged and negatively charged amino acids. This classification dictates exactly how they're going to interact with water, and crucially with each other when they start folding up. Section 3, primary structure and backbone. Okay, let's start linking these individual building blocks together. Amino acids connect end-to-end to form a polypeptide chain, and they actually lose a water molecule in the process to form a peptide bond. Because of a neat chemical property called resonance, where electrons are shared across atoms, this bond has a partial double bond character. So what does that mean for us? It means it absolutely cannot rotate. It creates a very rigid, planar, and uncharged backbone that holds the entire chain together. And because of this rigidity, the peptide bond basically has to pick a lane. It almost always adopts a trans configuration, where the bulky alpha carbons are sitting on opposite sides. Why? To avoid a molecular traffic jam. If they were on the same side, in the cis configuration, their atoms would literally crash into each other. Chemists call these atomic collisions steric clashes. The one major exception here is proline. Its unique ring structure actually allows for the cis form sometimes. All right, so the peptide bond itself is locked in place, but a protein obviously still needs to fold, right? It manages this because the single bonds right next to the peptide bond do allow for rotation. We measure these rotations as phi and psi torsion angles. The phi angle is between the nitrogen and the alpha carbon, and the psi angle is between the alpha carbon and the carbonyl carbon. It's the freedom of these 2 specific angles per amino acid that gives the chain the flexibility it needs to twist into complex shapes. And that brings us to the famous Ramachandran plot, which beautifully illustrates the actual boundaries of that flexibility. You see, while those bonds can rotate, they can't just rotate anywhere they please. This plot proves that roughly 3/4 of all theoretical angle combinations are physically impossible because of, you guessed it, steric clashes. Atoms just cannot occupy the exact same space. And honestly, this is a good thing. This restriction wonderfully limits the folding possibilities, which helps the protein rapidly find its unique, stable shape without having to randomly search through infinite configurations. Section 4. Secondary structure folds. Now let's see what happens as local hydrogen bonds start pulling this linear chain into regular, repeating patterns. In nature, the two most common periodic structures you're going to see are the alpha helix and the beta pleated sheet. The alpha helix is a tightly coiled right-handed rod. Here, a hydrogen bond forms precisely between an amino acid and another one situated exactly 4 residues ahead of it. The beta pleated sheet, on the other hand, is completely different. It's formed by fully extended strands lying right next to one another, linked by hydrogen bonds in either parallel or anti-parallel directions. But, not every amino acid likes being twisted into a tight coil. Certain ones actually act as helix breakers or destabilizers. Proline is the classic troublemaker here. Its rigid ring structure is missing a necessary NH group for hydrogen bonding, so it just breaks the coil. Other amino acids like valine or isoleucine are simply too bulky and cause those steric clashes we talked about. And then you have ones like serine or aspartate that have side chains that actually compete for the hydrogen bonds, which messes up the regular pattern. Now, to keep the protein from just turning into a long, straight needle, the chain has to fold back on itself to become compact. It achieves this through reverse turns, also known as beta turns and loops. These force the chain to abruptly change direction, almost like a hairpin turn on a mountain road. A crucial thing to remember is that these turns almost always lie on the outer surface of proteins. They are perfectly positioned to interact with other molecules and the watery environment of the cell. Section 5, Tertiary and Quaternary assemblies. Let's finally look at how all these coils, sheets, and turns pack together into the final, fully functional machine. The overall 3D shape of a single polypeptide chain is called its tertiary structure. Take a look at a compact protein like myoglobin, for example. This highly scrunched up shape is primarily driven by something called the hydrophobic effect. Since the inside of a cell is an aqueous, watery environment, the protein actively folds itself to hide its nonpolar, water-hating amino acids deep inside a central core. That leaves all the water-loving, polar residues safely on the outside surface. But for a lot of proteins, the tertiary structure isn't actually the end of the story. Many functional machines need multiple folded chains, which we call subunits, to link up and assemble into a massive quaternary structure. Sometimes it's just a simple pair, but other times you get incredibly complex structures, like virus coats made of dozens of repeating subunits all working together. A really classic example of a quaternary structure is hemoglobin. It's a tetramer, which just means it is made of 4 distinct subunits, 2 alpha chains and 2 beta chains. And they don't just sit there stuck together like glue. Subtle dynamic shifts in how these 4 subunits arrange themselves are exactly what allows hemoglobin to pick up oxygen in our lungs and then efficiently release it into our tissues where we need it. So let's do a quick recap of how we build these amazing biological machines. Step 1, we link the amino acid sequence together into a primary structure. Step 2, the backbone forms local hydrogen bonds, giving us secondary structures like helices and sheets. Step 3, the whole chain folds up to hide its hydrophobic parts, creating the tertiary structure. And finally, step 4, if the protein needs it, multiple folded units will assemble into a quaternary complex. We've seen just how incredibly precise this folding process has to be. In fact, when you realize that swapping out just a single amino acid in this sequence can cause devastating health conditions like sickle cell anemia, it really makes you stop and think. What does this reveal about the breathtaking precision of molecular evolution? Every single sequence operating in your body today is the result of billions of years of refinement. Thank you so much for joining me on this tutorial, and I really hope this leaves you with a profound appreciation for the magnificent biological machines working inside all of ourselves.